the organizers of Resonance 25 for the opportunity. So I was a student who did BSc in Physics, Chemistry and Maths in 97. Not obviously here, but in the Residency Road campus. And I'm going to tell you a little bit about some of the stuff that our lab is interested in and take you through what is the mind of the worm and tell you why it might be interesting to study an organism that's very different both in looking and in dealing with whatever it's given in comparison to humans and other vertebrates. So, in order to think, in order to walk, talk, move your head, everything requires functioning in the brain. The brain is made up of a large number of cells which are called neurons, and these neurons essentially talk to each other. And the brain has so many neurons, each of them makes many, many connections with many different neurons. Hence, there's a lot of chatter that's essentially happening in the brain where multiple neurons are talking to multiple other neurons. So just to give you a very basic of how the neuron talks to another neuron, what we have is a presynaptic neuron that sits here, and that is conversing with a postsynaptic neuron. It's essentially talking to this postsynaptic neuron. So how does it do that? You have an action potential that's coming along the axon of a neuron. So you have the cell body of the neuron here. This has an axon that comes like this, and this is the end of the axon. Okay, this is the growth cone, or the one end of the axon. And as the action potential comes through, calcium enters. This then allows for neurotransmitters to be released from this presynaptic neuron. The neurotransmitter then binds to a receptor or another molecule, a protein that sits on the surface of this postsynaptic neuron, the neuron that it's going to be talking to. This causes changes in this postsynaptic neuron. And essentially that is how we do everything. In the final, so all the neurons that sense the environment give signals to a next level of neurons. Those in turn give more signals to another set of neurons, which then affect the muscle. So finally, whatever you're doing actually affects the postsynaptic muscle, which is which gets synaptic connections from a presynaptic neuron. Okay, that's essentially how we do everything. Now, a lot of work has already been done, which tells us that what neurotransmitters are, what postsynaptic receptors are, how these signals are transferred. But what we know is not complete. It's far from complete. And some of the questions that we don't know the answers to are who are the proteins that are sitting on the presynaptic neuron that allow for this process to happen? Who are the proteins that are sitting on the postsynaptic cell or the postsynaptic muscle or postsynaptic neuron that allow for this signal to get transferred via the receptors downstream onto this neuron? Okay, so all of these questions are things that we're very interested in understanding in our lab. And to do so, we essentially break it down using a small model organism. So why do we use a small model organism, which is essentially the worm? It's called Caenorhabditis elegans. This short answer is essentially our human brain has about 100 billion neurons, okay, and more than 10 trillion synapses. That number is really, really large. So trying to study 100 billion neurons and 10 trillion synapses becomes daunting in many, many ways. However, the human brain has around humans have around 25,000 genes that code for proteins. C. elegans, on the other hand, has 302 neurons, 7,000 synapses. We know where every single neuron in this animal is. We know where every single synapse in this animal is. But it has 20,000 genes, which is very similar to the number of genes that are seen in the vertebrate counterparts. So you can imagine that many of the genes that are required for making neurotransmitters, that are required for making the receptors, making the proteins that allow for normal neurotransmitter release, all of those are conserved in C. elegans, So this is a nice system to start understanding molecularly what these proteins are doing and how they might be affecting function. So just to give you a very brief overview on about C. elegans, all you need is a plate which has agarose like any other bacterial plate. You need some bacteria because worms feed on bacteria. And you need a microscope which has light at the bottom and light at the top, which will allow you to see these worms. And this is how these worms will look under this microscope. So it's very, very straightforward to work with this. It's not that difficult. And the life cycle of this worm is essentially 3 days. You go from egg to the hermaphrodite. The worms are hermaphrodites and males. So the hermaphrodite essentially keeps giving a clonal progeny, and the male on mating can allow you to do crosses and put different genetic backgrounds in the same animal. So in 3 days, you go from this egg to this hermaphrodite that's giving many more eggs. Making it a fairly simple system because in 3 days, it's not very long, right? You can do lots of these crosses, get whatever you want and figure out what you want fairly quickly. Apart from that, the other advantage is that it's completely transparent. So what we can do is we can tag any protein of interest to green fluorescence protein or GFP or red fluorescence protein, any color that we want, and we can very easily see it under a microscope. Here you have a protein that's been tagged with GFP, and you can see very nicely expressed in a large number of axons. It's seen in neurons in the head, so you can very easily look at it. Okay, making it a very nice system essentially to look at what's happening with respect to molecules or proteins. Our lab is interested in understanding two things. So one of the things that we focus on is looking at how cell adhesion molecules, those are essentially molecules that sit here or sit here, how they affect the neuromuscular junction. Where the neuromuscular junction is essentially where the neuron speaks to the muscle. So what we want to do is who are the molecules that sit, find out is who are the molecules that sit here and who are the molecules that sit here. How do they actually talk? How do they actually enable for normal functioning of this neuromuscular junction, and what are they actually doing sitting where they are? The other aspect, and this is what I'm going to talk about today, is essentially looking at how locomotion happens in this worm. And why are we interested in locomotion in this worm? Although locomotion in a worm is very different from human locomotion. They don't have hands, they don't have legs, nothing. It's very, very different. However, circuits that are required for locomotion are conserved. The molecules that are involved in these circuits are conserved. The neurotransmitters that are required for talking between neurons are conserved. So we essentially look at how locomotion happens and who are the molecules that are involved in the process of locomotion. And today I'm going to be telling you a story about drunk worms and dopamine and what dopamine does to enhance being drunk in worms. Okay? So what's dopamine? Or many of you might have heard about dopamine. It's a biogenic amine neurotransmitter. And in mammals, dopamine is known to regulate many functions, including emotion, coordination of locomotion, and cognition. Lots of functions require dopamine. There are many dopaminergic neurons in our brain which allow us to go through a lot of these processes. In C. elegans, on the other hand, there are H neurons. H neurons that give out dopamine. 6 of them are sitting in the head and 2 of them in the tail. You'll only see one each because the other is a pair which is on the opposite side. So you only usually see one on any given plane. So you have 6 neurons sitting in the head here and 1 pair of neurons. Just a little bit of an introduction to how dopamine functions. So you have a pathway from tyrosine, which is an amino acid, through dopa and dopamine, which is put into vesicles. These vesicles then go and fuse at the membrane and allow for release of dopamine. Okay? And apart from molecules that are required for release of dopamine, you also have a receptor that sits on the neuron that has dopamine, and this is thought to allow for maintaining the dopamine levels from this neuron. Okay? Now, once the dopamine is sent out, it's very similar to what I said before. It binds to the receptor that's on the postsynaptic neuron. The dopamine binds to this receptor and then it activates the neuron that is postsynaptic. And this process continues, and this is required for pretty much everything that we do, from locomotion to memory. Okay. So now the next question is, does alcohol have any effect on this neurotransmission? And is there any way by which alcohol might affect this? Now, if any of you were to see somebody drunk walking around, you will see that they are not very coordinated, right? I mean, most of you must have seen that. They kind of stumble along. And that is because a lot of the neurotransmitters are affected in the presence of alcohol. So too much alcohol causes a lot of different changes in neurotransmission. And a lot of previous work has shown that different neurotransmitters like glutamate, GABA, and dopamine are all intrinsically linked with alcohol and could be affected by alcohol. Further, they could also affect alcohol levels. So what is known? What is the link between dopamine and alcohol so far? What we know from literature or from humans, from studies on humans? We have very, very contradicting data. Data is taken from different parts of the human brain, from mice, from other model systems, and a lot of the data is very contradicting. Some of the data essentially indicates that if you look at the brain of an alcoholic, you see less dopamine. You see less amounts of molecules that are required for releasing dopamine. You see less amounts of dopamine receptors. All of it is less in an alcoholic brain. But there is also data that indicates that in the presence of ethanol, the amount of dopamine released is actually more. So we wanted to see if there was a link between dopamine and alcohol, and we wanted to use the C. elegans, or the worm, as a system to address this question. Okay, and what we decided to do was, we essentially went through literature and we found that there's a very simple assay to look at drunk worms. All you need to do is put the worms on 400 millimolar of ethanol and see what happens to the worm. This paper, Davies et al. In 2003, essentially described very nicely what happens when you put a worm on alcohol. So what they showed was that when you put the worm on alcohol, initial 10 minutes, it's fine. It's moving around absolutely fine. From the 10th to the 15th minute, every single worm in that plate paralyzes. Every worm paralyzes, and just immediately paralyzes. You will see that paralysis really easily. It just paralyzes. After around 35 to 40 minutes, they start recovering. That is, now they become desensitized to alcohol. They are able to use, that alcohol doesn't seem to affect them anymore, and they're able to move around fairly happily after being desensitized to alcohol. And then when you look at the worms, after 1 hour or 2 hours, they're fine on that alcohol is not affecting them anymore, and they're moving around absolutely fine. So what we wanted to do was, we wanted to look at every molecule that is in this dopaminergic pathway and see how alcohol might be affecting the animal when we remove each of these molecules or each of these proteins. So we did the screen, we removed each of these proteins, and we essentially found that most of them were fine. Most of them showed no visible phenotype. We were not able to differentiate between the control wild type animals in most of the cases, except in one case. And I'll tell you about this molecule in a little while, but the molecule that we found that had a defect is called doped. Okay? And you'll see what the defect is. So every single animal, we see this defect. So essentially we looked through the entire 2 hours to see what the defect is. Initially we found no defect, but when we looked at these worms after 2 hours, we found a very, very obvious defect. So these are wild type worms, and you can see that they're essentially moving around happily after 2 hours of ethanol treatment. Nothing seems to be the problem with them. And these are the mutants. I'm going to tell you what these mutants are. They're called DOPE2 mutants. And you can see that essentially this animal is not able to move. It's trying its hardest to pull itself in front, where the anterior part is kind of allowing it to move, but the posterior is just not allowing this animal to move. So it essentially goes around in circles, where each circle takes hours and hours and hours to complete. While this animal is very happy, it moves around very, it has no problem. So then we thought, okay, how do we actually look at this defect? Can we actually quantitate it? Can we actually see what is the defect? To do that, what we did was we essentially found out how many times the posterior of the animal is moving and how many times the anterior of the animal is moving. In compared to wild type, where the anterior is moving around 20 times per animal and the posterior moves around 15 to 16 times, these mutant DOP2 animals showed a huge defect in this movement. There's less anterior and less posterior movement. In fact, posterior movement is almost nullified, like you saw in the video. We also looked at the amplitude, that is the distance between, if you draw a line across the midpoint of the worm, you just find out the distance between the highest point and this line. And again, we found that that is also much less in these animals, which is not surprising. You saw that they could hardly move. So we did this with everything in the dopaminergic pathway, and we found that only these DOP2 mutants show this very, very dramatic phenotype. This is a molecule that is required to sense the ethanol. So if you cannot sense ethanol, everything is fine. The worm moves a lot in the presence of ethanol. We just use this as a control. This is called slow one. We use it as a control and that behaves perfectly fine. But otherwise, we found that only this showed a very stark phenotype. And this was the only thing that when we looked on the plate, we could easily see this phenotype. So the next question was, this is all very well and good, but we need to have controls, right? We don't know whether in the absence and presence of ethanol, wild type animals are behaving normally. We don't know whether these animals, DOP2 mutants, actually have a phenotype in the absence of ethanol. So we did all of those controls. So you can see wild type in the absence of ethanol moves very similar to how it's moving in the presence of ethanol. Again, all of these graphs are going to be just number of body bends and the amplitude of body bends, okay? And you can see that wild type ethanol plus and minus ethanol, it looks perfectly fine, exactly the same. Then we looked at DOP2 mutants in the absence of ethanol, comparing it to wild type in the absence of ethanol, and it looked very much fine. There was no problem. It's able to move absolutely fine in the absence of ethanol. So something in ethanol is what is causing this. So then we thought, okay, let us also look at other muscles that could be affected by ethanol and see if there are any defects. And we looked at the muscle that's required for taking in food, the pharyngeal muscle that keeps moving in and out, that allows for food intake. And we found that these muscles were not affected at all. So this seems very specific to the posterior locomotion. That is what is giving rise to this phenotype. So the next thing we wanted to do was, now we have this phenotype, but in order to confirm that it is this mutant that is giving this phenotype, we have to put back this protein in the animal. So we put back this protein in all those 8 neurons that express this protein. And we found that it went back to looking just like the wild type control animals. So you can see both the number of body bends and the amplitude went back to being just like wild type, indicating that it is this protein. So you remove this protein, then it becomes more sensitive to ethanol. And if you put back this protein, you get back your wild type or control animal phenotype. Okay, so far, essentially you have this DOP2 which sits in the dopaminergic neuron, and in the absence of this DOP2, you're getting the worm to just move around in circles. It's hardly able to move, and it's giving a lot of problems to this worm, essentially. So the next question we asked was, does this molecule actually affect dopamine levels? So DOP2 as a molecule is essentially a receptor. It sits on the presynaptic neuron, the neuron that's actually sending out dopamine. That's the neuron where DOP2 sits, and nobody knows what it does. So what we think is happening is it's somehow regulating dopamine levels. So that is our hypothesis. Our hypothesis is that this is somehow regulating dopamine levels. Now, how can we actually figure out if this is correct or wrong? So one experiment we can do. So if it is regulating dopamine levels, you can think about it 2 ways. It is loss of this molecule causes less dopamine. Loss of this molecule causes more dopamine. It's one of these two, right? Regulating some level of something means it's either giving more of this or less of the same. So we thought, okay, let us do this systematically. Let us first look at something which doesn't have dopamine. Now, if this is actually causing less dopamine, then you can imagine that if you lose dopamine altogether, you will see exactly the same phenotype, exactly what you see with these DOP2 mutants. So we took a mutant that is required to make dopamine. CAT2 is a tyrosine hydroxylase which converts tyrosine to L-dopa. So if you don't have CAT2, you have very little dopamine in the system. So in the absence of dopamine, how does the worm move? The worm is absolutely fine. It moves around very, very happily in the presence of ethanol. So it looked like what we see is not loss of dopamine. It could be maybe there is more dopamine. That is what is allowing this to happen. And how did we do that? We did a very simple experiment. We gave the worm dopamine and we also gave the worm ethanol. So we gave it both alcohol as well as dopamine and we wanted to see what happens. So this is just a worm with just ethanol, wild type worm with just ethanol. We already saw that it moves fine. This is a wild type worm without ethanol, with just dopamine. And we see again that it moves around fine. This is a wild type worm which we've given ethanol and dopamine. So this is not the mutant worm, it's wild type, it has dopamine and ethanol. And you can see that it completely mimics the previous phenotype. It's not able to move. Indicating that what we might be giving, the loss of this DOP2 protein might actually be causing more dopamine to come out. Right? So this is just the statistics for what we saw earlier. Wild type worm is just ethanol is normal. Wild type with dopamine is just normal. But if you put dopamine and ethanol, it behaves like the DOP2 mutants. So now we think, okay, it could be because there is more dopamine. So can we test it by any other means? Is there any other way by which we can actually see if there is more dopamine? Okay. And to do that, what we essentially did was we put dopamine and we put a tag onto the dopamine. Then we look to see how much of this fluorescence is there in the animal. Okay, just a very simple fluorescence assay. And you can see in these mutants, DOP2 mutants, with ethanol, there is much more fluorescence. So this indicates that there is indeed more dopamine in these mutants, and that is why they're showing this phenotype. So essentially, we have so far that the normal movement of C. elegans in ethanol seems to be dependent on this protein called DOP2, and DOP2 appears to release more dopamine in the presence of ethanol. So if you look at it schematically, you have DOP2 sitting in dopaminergic neurons. You remove DOP2, you get more dopamine. And I think that essentially all our data so far suggests that that is indeed the case. You remove DOP2, you get more dopamine in the presence of ethanol. Now the next question comes, what is the neuron? I told you that there are 8 neurons in C. elegans that have dopamine. 6 of them are in the head and 1 is in the tail. So which is the neuron that is required for this? Is it all the neurons? Or is it some permutation and combination of, or some subset of neurons that are required for this? So when you saw the initial videos of these mutants, one thing you might have appreciated was the posterior part seemed to be very badly affected compared to the anterior part. Right? The anterior was a little bit better than the posterior. So we thought, okay, let us see if it's the posterior neuron, because that to us made maximum sense. And how did we do this experiment? We just killed this neuron. We killed this one neuron, 2 neurons, because everything is in pairs as well. We just killed this neuron in C. elegans. So you have this neuron, you have DOP2 here. If you don't have this neuron, then there is going to be no dopamine release. The neuron is dead, everything is gone. So whatever phenotype we saw should be completely lost. And that is essentially exactly what we see. So this is wild type, which has not got that neuron killed. When we kill that neuron in wild type, it looks very similar. It looks just like wild type. In the mutant DOP2, we already see a phenotype, and when we kill that one neuron, you completely lose that phenotype. It goes back to being just like wild type, both for the body bands as well as the amplitude. So just to show you some of the videos, this is wild type where we've killed the neuron, and it's able to move. It's not very happy because killing these neurons always affects the worm in some way or the other. This is DOP2, which we have not killed any neuron. It's fine. It's just trying to move around, but it's not able to do anything. Then we just took these animals, which could not do anything, and we killed that one neuron. And you can see that now it's able to move. Not, I mean, again, just like wild type, it's not as fast as the normal one, but it's able to move very happily when compared to these guys. So what is this? What does this show so far? It shows that DOPE2, which sits in this posterior neuron, PDE neuron, seems to be functioning to allow for increased dopamine levels. Right? So you remove this, you remove DOPE2 from PDE, you're getting more dopamine. So now we come to a point where, okay, we have this neuron that seems to be having this DOPE2 receptor that allows for high levels of dopamine to be secreted. Now the question is, can we actually find out why this? What is the end point? Because at the end of the day, it is the muscle that drives the worm around. So how is this one neuron affecting the muscle? To do that, we thought a little bit about this circuit. Okay, so all systems that have a number of neurons essentially have a set of neurons which are called sensory neurons. These allow people to sense what is in the exterior. These neurons, synapse, make connections, essentially talk to a second set of neurons which are called interneurons. Okay, so these neurons talk to interneurons. Interneurons then talk to a set of neurons which are called motor neurons. Motor neurons are the neurons that talk to the muscle. Okay, so this is a very simplistic pathway which is seen in any system. It's exactly the same path. So motor neurons in humans send out a neurotransmitter which is called acetylcholine. In worms also the same thing. The motor neuron sends out acetylcholine. And this acetylcholine is seen by the muscle and allows the muscle to contract and allow for normal movement of the worm. And the same thing happens in us. Acetylcholine is released and it allows us to contract like this. Any kind of contraction requires this acetylcholine. So what we thought was, okay, so we now have to see what is it that is affecting, how is it affecting this motor neuron and hence affecting the muscle, right? So this becomes very important. How is something that's sitting here at the level, the neuron that I talked about is a sensory neuron, how is this guy somehow affecting this? So in order to do this, we first decided, okay, I told you acetylcholine is what is sent out from here. Acetylcholine is what is sent out from here for us. Acetylcholine is what is sent out from motor neurons in worms. So we looked at every component of this acetylcholine system. So our hypothesis was, what is happening might be that there is less acetylcholine. So if you remove any component of the acetylcholine system, you will have less acetylcholine. Will that mimic this phenotype? We looked at every component of this acetylcholine system and none of them showed a phenotype. All of them were like wild type. There was no difference at all. Then we thought maybe what is happening is it is not causing less acetylcholine. It is allowing for more acetylcholine to be there. And hence the muscle is contracting more and that is preventing the worm from moving properly. So in order to do that, we thought, okay, how do we devise and have an experiment that will allow us to mimic more acetylcholine? So this is just to give you a very brief understanding of this motor neuron. This acetylcholine neuron sends out acetylcholine. Acetylcholine binds to acetylcholine receptor that sits on the muscle and allows for muscle contraction. Apart from all of this, you also have an esterase. You have an enzyme which is called an esterase. The esterase sits at the synapse and it breaks down excess acetylcholine. So now if you want to have more acetylcholine, can you just break down the esterase? If you get rid of the esterase, you will have more acetylcholine. That seems very simple. So there's a drug available in the market which is called Idica. This inhibits the activity of the esterase. So if you add this drug, you have too much acetylcholine. Now we first want to test that this is fine, that it is indeed the case. Now one can imagine that if you have too much acetylcholine, there is going to be too much muscle contraction. If there is too much muscle contraction, the worm eventually just paralyzes because it's just you can just imagine if your muscle kept contracting, you will eventually it'll just give way. You'll just have an active paralysis, and that is indeed what you see. You add this drug at 0 time point, nothing happens. By 120 minutes, all the worms are paralyzed. So we thought, okay, let us take a nice 60-minute time point where few of the worms have started paralyzing and see if we can mimic this. And then put the worms onto a plate which has ethanol. And we use wild type worms, not the mutant worms which already show this phenotype. We take wild type worms which are completely normal, try and get more acetylcholine and put them on a plate with ethanol. And you can see what happens. So So you put a lot of acetylcholine, this worm has a lot of acetylcholine. Again, the anterior part of the worm is trying to move, but the posterior is just not cooperating. So it's essentially just dragging itself and trying to move on the plane. All of these videos were made for the same amount of time, which was approximately 1 minute for every. Okay? So you can see again, it's just not able to move. So then we thought, okay, this is all very nice. It could be acetylcholine. Then if we want to show that it is definitely acetylcholine, if we remove some component that is required to make acetylcholine, that will reverse this phenotype. Right now there is no acetylcholine, so it won't give this phenotype, and that is indeed the case. So you have a choline acetyl transferase that is required to convert acetyl-CoA and choline to CoA and acetylcholine. So if you remove choline acetyl transferase, you will have no acetylcholine. And will that then affect the worm? How will the worm be then? We see that it looks exactly like wild type. So this is wild type without aldicarb on ethanol, it looks fine. And you remove and you put aldicarb that you have too much acetylcholine, you see the phenotype that we saw earlier, and you remove choline acetyl transferase and you lose this phenotype. So indicating that the end point so far seems to be that there is too much acetyl. The phenotype seen in these mutants is due to increased acetylcholine levels. So you have essentially what this neuron is, a sensory neuron, and this neuron on losing this DOP2 protein seems to be causing increased dopamine release, which in turn seems to be causing increased acetylcholine release at the neuromuscular junction. Now, the question we next wanted to ask was, how is this happening? What is the circuit? There must be a circuit to allow for this, right? Because I'm saying sensory neurons. Sensory neurons are very far from the muscle, and they are somehow affecting the motor neurons. So who is this circuit, and what is the circuit that's allowing for sensory neuron to affect the motor neuron? So what we knew from literature was that this sensory neuron PDE makes many connections with this one interneuron which is called DVE. So a lot of previous work from multiple groups has shown that muscle contracts, this neuron takes in calcium, this neuron then gets activated. And now I'm going to move very slightly to a different concept. Instead of sending a neurotransmitter that I've talked about so far, this sends out something that's called a neuropeptide. And you don't have to worry about this term neuropeptide. Neuropeptide is essentially just a small protein that behaves like a neurotransmitter. Okay? I'm just going to call it neuropeptide. You can just think about it as some other transmitter. This neuron sends out this neuropeptide. The peptide then binds to the receptor, very similar to a neurotransmitter, binds to a receptor on this neuron, activates this neuron, and allows for acetylcholine to release. So what we thought was this sensory neuron synapses on this neuron and through dopamine, and this then allows for this pathway to happen. So one can imagine that in this scenario, if you remove DOP2, which is required for too much dope, which when you remove you have too much dopamine, this then allows for too much of this neuropeptide to be sent out, and this then allows for too much acetylcholine to be released. And this is what is giving rise to what we see where the worm is unable to move. So if we want to actually show this, we can show it at different levels. I'm just going to give you a couple of examples of what we do. So what we thought was, if this is indeed the case, then if you don't have DOP2 and you don't have DOP1, what will happen? DOP2 has to go, dopamine has to be seen by DOP1 here. So if you don't have DOP1 here, you will lose this phenotype. It will go back to being wild type. And then you can do a very clever experiment in worm. You can put back DOP1 just in this one neuron. In essentially this one neuron, you will get back what you see earlier if we think this pathway is correct. And that is essentially the experiment we did. So you don't have DOP2, you don't have DOP1. And what happens when you don't have DOP2 and DOP1? You lose this phenotype, that is DOP2. DOP1 does not have a phenotype, and you go back to having exactly wild type like phenotype, no phenotype, essentially, right? So now, in this system, if we put back DOP1 just here, can we get back the DOP2 phenotype? Because DOP2 is still not there, right? So if you put back dope 1 here, the dope 1 will see the dopamine, everything will happen properly. We'll have too much acetylcholine. And that is exactly what happens. We put back dope 1 into that one neuron and it just goes back to looking exactly like dope. Right? So indicating that this is indeed the circuit through which this process is happening, where we have this neuron, it's sending out too much dopamine in the absence of dope 2. It's seeing dopamine receptor, which is sitting on this neuron and allowing for too much neuropeptide to be released. So right now what we're doing is we're doing more experiments by looking at seeing if the peptide, the next question was if this is indeed the case, what we would think is that if you have too much neuropeptide being released, that would also show exactly this phenotype. If you overexpress this neuropeptide, you have too much neuropeptide, that would again cause too much acetylcholine to be released, same phenotype, and that is exactly what we see. When we take 3 independent lines where we have too much peptide, it looks exactly like the dope 2 phenotype. So right now what we're doing is we're doing experiments to look at all the other components to see if exactly what we think is happening is actually happening or is it going through some other mechanism. But so far what we think is happening is that in this PDE neuron, in the absence in the PDE neuron, in the absence of dope 2, there is too much dopamine being released. This then binds to dope 1, that is a dopamine receptor sitting on this interneuron called TPA, which sends out the neuropeptide, which then binds another receptor here and allows for acetylcholine. And why is this interesting and why is it significant? Previous work in different systems, including Drosophila, has shown that in mutants where you have too many acetylcholine boutons, okay, so this is essentially a cholinergic boutons, they become much more sensitive to ethanol, which is exactly what we see. That the end point of having too much acetylcholine makes the organism more sensitive to ethanol or is not able to recover from too much ethanol like wild type animals are. So with that, I'm going to end and I'd be very happy to take questions, but I just want to thank the people who've done this work. This work was essentially done by graduate student Anuradha and a postdoc Pratima. And the experiments where we did the ablation, where we killed a single neuron, was done with Anandya Ghosh Roy and Harjot Kaur at NBRC. And the plasmids, the reagents were from CGC and Adgene. We thank a number of labs. Some of it is gone from here, I don't know why. I also want to thank funding agency India Alliance, IISr Mohali for generous funding, SERB, DSC SERB also for funding, and I'm currently at IISc Bangalore, and if any of you want to come by and look at worms, you can come to IISc. And with that, I want to thank you all for your attention. I'm happy to.